This is audio assignment 7-1, Introduction to Clinical Microbiology. Microbiology is the study of organisms so small that they can only be seen using a microscope. In the clinical side or medical microbiology, we study viruses, fungi, bacteria, and parasites that cause disease in humans and other animals. In your larger hospitals or reference laboratories, each of these specialties may be in a separate department of the lab, but in your smaller laboratories or physicians' offices, or POLs, only the less complicated procedures are performed, and most parasitology, virology, and mycology specimens are sent to a reference lab. So, before we start to discuss bacteria and other organisms studied in microbiology, we do have to know about the system we use of naming things or nomenclature. All living things are classified according to international rules of nomenclature. It's called taxonomy or binomial nomenclature, and binomial means two-part names. Now we have been classifying things into groups since the beginning of time. But I do want to talk about Carl Linnaeus historically. He's a botanist from the 1700s, and he authored this original binomial nomenclature, and he used simple physical characteristics to identify and differentiate between species in order to form a method of classifying the organisms. So he developed a hierarchy or type of pecking order of groups, and each of those groups can be subdivided into other groups. This type of classification provides scientists a way to sort and then group organisms for easier study. So, as scientists have reorganized classifications and based on new discoveries and information, in 1990, Carl Woese added a new category or domain called the archaea. Until that time, biologists had taken for granted that all life on Earth belonged to one of two primary lineages, the eukaryotes and the prokaryotes. The eukaryotes consist of animals, plants, fungi, and certain unicellular organisms such as paramecium. That EU prefix means true and kary means nucleus. And then the prokaryotes, their domain is the bacteria and archaea. Uh, the prefix pro or pro means before, and as I say, kary means nucleus. And I just want to, as a side note, organisms that fall within the domain archaea, they live in the most extreme environments on the planet Earth, such as Yellowstone, for example. So, as I said, species of organisms are given binomial names. The first part of the name is the genus to which the organism belongs and is written with the first letter capitalized. The second uncapitalized name is the specific epithet or specific description of that genus. Scientific names of organisms are underlined when handwritten and italicized in print. And in this slide, I'm using the bacteria Staphylococcus aureus, and it's abbreviated S. aureus. We would never leave out the genus part if we were abbreviating. We'd always have the Staphylococcus in there. And just to show you, Staph is the genus and aureus is the species. Many bacteria have multiple species within their genus. So there's Staphylococcus aureus, there's Staphylococcus epidermidis, there's Staphylococcus saprophyticus. So there are other types or children within that naming. You can kind of think of genus as the parent, and then the species are the children that follow. Nomenclature or naming systems for bacteria, fungi, and parasites all follow these rules. And because viruses are not living organisms, they do not follow the system of nomenclature. Viruses are named after the disease they called, or, excuse me, the disease they cause. They're named after for the organism or the organ they might infect. They're named after the place where it was first found, or it can be named after the person that discovered it, or just after people in general. There's some general characteristics of bacteria. First of all, bacteria are single-celled organisms that multiply by fission. And fission is where the parent body splits into two separate cells to form colonies. And you hear the word colonies, that's a term used when bacteria is observed to be growing, so for example, in a petri dish. There are three shapes to bacteria. It's called the bacterial morphology. You have your coccus, which is round, the bacillus, which has a rod shaped. You see in the middle picture on the slide. And then you have spiral shaped bacteria. These types of bacteria that you see in box C is a fluorescent picture. This type of bacteria or species of bacteria, it will not absorb a general stain, so you have to use a different staining process for that one. So we report these gram stain reactions as gram positive or gram negative. And gram staining is a microbiological procedure that categorizes bacteria based on the physical and chemical structure of their outer surface of the cell or the cell wall. Gram-positive bacteria have a thick layer made up of polymers of protein sugar molecules called a peptidoglycan layer. Gram staining of the peptidoglycan layer with a chemical called crystal violet results in a purple coloration of the gram-positive bacteria. Other chemicals are used during the staining process, but it's the crystal violet that's retained by the cell wall, and that causes it to keep the purple color. So gram-positive bacteria are purple, and you see in box A an example of a gram-positive cocci. Gram-negative bacteria, circle B, have an additional outer membrane whose properties resist the chemicals in the beginning of the staining process, and the result is the loss of the crystal violet stain. We use a counter stain that stains these bacteria red or a pink color. This gram stain reaction is referred to as gram negative. Bacteria also have certain nutritional growth requirements. They need oxygen to live, for example. And just some examples of categories, I guess you could call it, of growth requirements. We'll use the term first, fastidious, Fastidious. It's in your reading. You can also look in your glossary at the beginning of chapter for these definitions. So if an organism is referred to as fastidious, that means it has complex nutritional requirements. If it is an aerobic organism, that means it has or it requires air to grow. If it's an anaerobe, it means it does not require air to grow. And there are some that might be referred to as a microaerophilic or just a reduced oxygen type of organism. They grow in about a 5 to 10% carbon dioxide environment. Most of the bacteria will grow on what's called a blood agar plate, which is the most common type of medium used in the laboratory. It is a tryptic soy agar, and it includes soybean meal, casein. Casein comes from milk and cheese. It has amino acids, and it also has 5 or a 10% sheep blood added to it, solution of sheep blood added. Blood agar is especially useful when trying to cultivate or trying to grow fastidious bacteria, and also for observation of hemolysis on the plate. And we'll discuss the types of hemolysis you see on blood agar plates in other chapters. Just some terminology you should be familiar with, and the first one is normal flora, and that means that is a type of organism that inhabits the human body and is present also on the surface of inanimate objects or non-living materials. Pathogen means that it's able to cause an infection. You have a non-pathogen. That means it is a bacteria that does not cause infection. And then we have opportunistic pathogens. And these types of pathogens are able to cause infection when conditions are right. So that means your immune system has to be depressed or you're sick for some reason and this pathogen can take over. Also in this chapter you have readings on parasitology. In parasitology, we look at blood and tissue parasites, and in here this slide is giving you three basic types. Also, we'll be looking at those topics on parasitology in Chapter 8. However, we don't cover much on virology, so just a little on virology for now. Viruses are studied in some of our clinical microbiology laboratories. Viruses are not living cells, and they can only reproduce or replicate by invading another cell, such as the human body. Uh, Living organisms both contain DNA and RNA, but virus can contain only one or the other. And DNA, if you remember, is deoxyribonucleic acid. It carries genetic information, is found primarily in chromosomes, and RNA is ribonucleic acid, is a nucleic acid responsible for protein synthesis, protein production. Um, as you can see, viruses are very small and they do require the use of an electron microscope to actually study their morphology or their shape. But virologists believe there are more than 400,000 different types of viruses, with over 5,000 types being identified. Um, in the clinical lab, we use viral cultures, we use immunoassays, and some simple rapid tests to identify the types of virus that's making a person ill. You can see on table 7-7 the types of tests using to identify different types of viruses. We'll use a couple of the tests when we get into immunology department. The term mycology means the study of fungi, and in those categories of fungi, you're going to find molds and yeast. Um, Molds exhibit a filamentous type of growth, kind of like maybe a dandelion, um, and then yeasts on a media have a pasty or a mucoid form of fungal growth, which we found more than 50,000 different types of valid species of mycology type of organisms. Okay, so some characteristics of molds. They do produce a mycelium is composed of hyphae. This is a mass or mat of hyphae forming the vegetative portion of the fungus. Um, and they form spores, which is generally the reproductive body of the mold. There's two types of spores, sexual, which means that they reproduce by fusion of nuclei, and then you have asexual spores, which are the most common, and these can grow into another cell without uniting with another cell. They can grow in another cell without uniting. Generally, two media is used rather than plated media. And there's a few reasons for that. For one, you don't want to release spores into the environment. So having tubed media means that there's less chance for spore release. There's less chance of dehydration. These do have to grow for quite a few long period of time, and the media that we use in the laboratory does dehydrate or lose its water. Plus, there's the ease of storage. So we usually will inoculate the agar that's in the tube using a straight line, just pulling it straight out, and we preliminarily will ID it on different growth patterns on the types of media. When we are going to identify different types of yeasts, some of their characteristics, you'll notice that they have um, a germ tube and they will usually bud. These are going to stain gram positive when you stain them, but this is a great picture of a yeast showing the mother cells and producing a daughter cell. Mycology, or excuse me, yeasts. I mentioned gram staining. There's going to be times when you may be gram staining an organism, or excuse me, gram staining a sample. Could be a urine sample, could be a culture from someone, vaginal culture, or along those lines. They will stain a gram positive, but they are larger than bacteria, so they're a little, they're easier to identify as a bacterial type, and also we can see the budding process that goes along with it. So when we identify both molds and yeast, there's a process that's used, and I'm just going to go through this very quickly. You don't have to write it, take any notes, but we do a direct exam and we look for spores, hyphae, mycelial elements, budding yeast, mycotic granules. We perform a wet mount. Those are also good for yeast. Um, we do a potassium hydroxide or a KOH prep. Potassium hydroxide prep is usually done on things like skin scrapings, hair, nails, sputum. KOH, the potassium hydroxide in it, will clear the specimen of tissue cells, mucus, and things like that so we can see the fungal elements. And then also we use stains to observe fungal structures. Any questions, be sure to email your instructor through Canvas.